the components of the adaptive immune system, and this is Lynn. Let's go ahead and get started. So the following learning objectives are: distinguish between the active and process forms of adaptive immunity and provide examples of each. We'll list the different types of B cells, T cells, and describe the function of each type. We'll describe the structure of BCRs, TCRs, and respective co-receptors. We'll describe the effector cell functions of CD4 T cells, CD8 T cells, and B cells. Define and apply the class terms isotype, subtype, allotype, idiotype, monoclonal, oligoclonal, polyclonal antibodies, FAB fragments, FAB2 fragments. Identify and describe the role of key domains of antibodies, including FC, antigen binding site, C prime binding site, and FCR binding site. Describe the quantitative and qualitative differences in adaptive immune primary responses and secondary responses when they encounter an antigen, and relate the differences to diagnostic serology. We'll define and apply the terms vaccination, herd immunity, adjuvant, booster, and conjugate. Describe the different types of vaccines, including the benefits and limitation, and provide examples. We'll describe the different components that are present in vaccine, and explain the challenges to vaccination and why vaccines can fail. All right, people, let's get started. So an overview of the adaptive immune system. So let's start with the adaptive immunity. The adaptive immunity involves specific lymphocytes responses to a particular antigen. An immune response is defined as when the immune system comes in contact with an antigen, there's going to be the lymphocyte proliferation. White blood cells begin to start to get active. This immune response consists of lymphocytes and their products. Typically, the acquired and adaptive immunity takes a lot longer time than the innate immunity, and there are two branches within the adaptive immunity. The first branch is called cell-mediated immunity. This involves T cells that will destroy target cells with the help of proteins also known as pephorins. These pephorins can literally penetrate the cell membrane or the target's membrane and cause the cells to either go through apoptosis or lyse. The antibody-mediated immunity involves the B cells. These can then differentiate into plasma cells that will disperse out antibodies, and that can then activate complement systems, which is also a series of proteins, as well as recruit other white blood cells to neutralize the target. On the right-hand side, there's a graphic that shows you how bacteria, viruses, and tumors can trigger innate immune responses. You can see that any of these are considered antigens, and that will then trigger by the release of chemicals to the environment themselves. It utilizes a cell called the antigen-presenting cell, APC. These will then take information to share that to the T cells, and T cells begin to either convert into the cytotoxic version on the bottom, or they can become the plasma cells and then begin the release of antibody. We'll take a look at that in a few slides. This is another graphic showing you the branches of T cells and B cells. The T lymphocytes are the ones that provide the cell-mediated immunity. They are able to go directly against a target, and it does require an antigen-presenting cell. We also have the B lymphocytes, which are going to be effective releasing the antibodies, and they do not require a specific antigen-presenting cell. You'll see over here there are two subclasses of T lymphocytes. The first class is called the cytotoxic T cell, and the other class is called the helper T cell. There are signals that are released throughout the immune system called cytokines, or also known as interleukins. These are not hormones, but they're localized signals that are noted by a number. They can recruit activity as well as alert nearby immune cells to become more aggressive. Antigen. An antigen is a substance that is different from the molecules of the body, and it binds to a component of the adaptive immunity. Antigens are usually proteins or large polysaccharides, for example, protein cascades of viruses, the cell wall of a bacteria, of fungi, bacterial toxins, and even fragments of cancerous cells can be considered as antigens. Self-antigens are the body's molecule. Typically, they do not bind with the body's immune component, except for in cases when somebody has an autoimmune disorder. The antigenic determinant is called the epitope. This is a specific site on the antigen that is recognized by different parts of the immune system. The immunogen is going to be defined as an antigen that induces an immune response. and its ability is to cause immune response called immunogenicity. It does increase based on the foreignness of the antigen, the size of the antigen, and of course the quantity of the antigen. Another term is called haptens. Haptens are substances that are just too small to function as an antigen by themselves. But when they're attached to a host molecule, they can become considered antigenic. An example could be a lipid toxin inside the poison ivy plant that can then penetrate the skin, and when it combines with the bodily protein, that will then elicit an immune response. On the right-hand side, you can see a picture of different kinds of antibodies: antibody 1, antibody 2, antibody 3, antibody 4, and you have the antigen in the middle. Different regions on the antigen will have a different epitope or determinant site that can interact with the white blood cell. Antigen-presenting cells and MHC molecules. MHC stands for Major Histocompatibility Complex. Antigen presentation is a display of the antigen on the cell's membrane surface. Right? You got to be able to alert your white blood cells to know what is foreign. There are two categories of cells that are able to present antigens to T cells. Number one, all nucleated cells of the body, and also the APCs, also known as antigen-presenting cells. Antigen-presenting cells, APCs, are any kind of immune cell that is able to specifically communicate the presence of that antigen to either a helper T or cytotoxic T. Some examples include dendritic cells, macrophages, as well as B lymphocytes. These are all considered APCs. Major histocompatibility complex, MHC. This is a specialized transmembrane protein found on the cell membrane that is required for antigen presentation. And there are two classes. The first class is called MHC class 1. These are presented on every single cell inside the human body, every nucleated cell, whereas the APCs displays both MHC 1s and the MHC class 2. So in other words, remember, MHC 1s is only found in all normal cells. MHC 2s are only found in immune-based cells. MHC class 1 molecules are glycoproteins that are genetically determined and very unique to every individual. They present a peptide fragment from within the cell to the plasma membrane. So the function of the MHC class 1 is to display a peptide fragment that is from the cell itself. So we call these endogenous proteins. In a healthy cell, the peptide fragments presented on the MHC class 1 molecule are considered self-proteins, and so they get ignored by the white blood cells or any kind of immune-based cells. When an infected cell, the antigen presented on the MHC class 1s are then considered foreign antigens. These ones will then get recognized by other white blood cells, like the cytotoxic T cells, to be destroyed. Proteins that come from infectious agents can be cleaved, and the proteasomes organelle inside a cell can break that down, ship that into the rough ER, combine that with the MHC class 1s, and then display those onto the cell membrane. Let's talk about the MHC class 2s. These are considered the professional antigen-presenting cell proteins. MHC class 2 molecules is again a glycoprotein that presents a fragment of antigens that have been engulfed, ingested by one of these APCs. APCs engulf what we call exogenous antigens. They come from the outside of the body, digest those peptide fragments, and display them onto the cell membrane. And this is called the exogenous pathway. When you display something that is foreign on the MHC class 2, it provides a mean of telling a white blood cell that this is the target. And the purpose here is to communicate with a specific type of T cell called a helper T cell, so that it can relay other signals to recruit other white blood cells for attack. On the right-hand side is a schematic structure of the class 1 MHC protein that's bound to a fragment. Notice that it has several parts. It's formed by the alpha 1 subunit, alpha 2 subunit, and it even has an alpha 3 subunit that is associated with the beta 2 subunit. And these two parts, the alpha 3 and beta 2, are close to the plasma membrane. On the top end, you'll see there's two protruding alpha helices, and there's a small groove which is specific to binding the antigen fragment. Lymphocytes are the key white blood cells of the adaptive immune system. We have T cells and B cells that have unique receptors that are composed of several different proteins that bind to specific antigens. Typically, you're talking about at least 100,000 receptor complexes per cell, and a receptor complex will bind to one particular antigen. The T cells are matured inside the thymus, whereas the B cells are matured inside the bone marrow. Lymphocytes that enter the bloodstream, for the most part, are going to go via the lymphatic drainage sites. And on average, only 2% of the body's lymphocytes are found in your blood. If you have a higher amount of this, that indicates some sort of an infection. T cells and B cells are basically almost indistinguishable. So on the right side, you can see a family chart of the T cells. We start off with the committed lymphoid progenitor cell. In the thymus, they will then branch down to the T precursors, either the helper T or the cytotoxic T. Helper Ts are subcategorized as Th1, Th2, T17, and T regulator cells. And then on the right-hand side, the B cells simply become either naive B cells, memory B cells, or plasma cells that can release antibodies. On the far right, you'll see that it can also become the NK cells, natural killer cells, and then on the left-hand side, it can also become the NKT cells. We're going to focus strictly on the T cells and the B cells only. T lymphocytes have unique receptor complexes, about 100,000 of these per cell. Each complex will bind to one specific antigen. TCRs, also known as T cell receptors, is an antigen receptor of the T lymphocyte. T cells have an additional protein receptor molecule called the co-receptor, which facilitates physical interactions with other kinds of APCs. The CD proteins is a category of co-receptors that have been studied. They're also referred to as cluster differentiation proteins. Helper T cells contain the CD4, and they are referred to as the TH cell or the CD4 plus cell. Same thing. The cytotoxic T cells contain the CD8 protein, so they're referred to as the T cytotoxic or CD8 cells. We also have memory T cells for both the T helper and also the T cytotoxic. The helper T cells, again, are divided to 4 subcategories. The Th1 cells are ones that are used to release a cytokine called IL-2. That's a general alerting-based signal. Th2 cells secrete IL-4 and 5, and their role is to alert B cells. Th17 cells are used to induce bacterial-based infections, and they produce IL-6 and IL-7 to alert neutrophils, which is one of the most common types of white blood cells that can engulf targets and antigens and pathogens. And then we also have the T-reg cells, T regulator cells that produce IL-10, which will dampen and reduce T-cell driven responses. These IL stand for interleukin. These are signals that are localized, strictly used inside the immune system for changing the ability for white blood cells to act. And then the last type of T cell is the cytotoxic T cell, which releases chemicals that will directly destroy cells that express a foreign antigen. T cell receptors. The receptors on T cells are made up of 2 polypeptide units, the alpha and beta. Hence, you'll see the alpha beta T cells. They form this heterodimer, which means there's 2 different parts. They recognize the portion of an MHC protein and the antigen fragments on it. Delta T cells is about 10% of circulating T cells and have 2 different polypeptides. They're only found inside the mucosal layer of the GI tract. The CD8 co-receptors on the surface of cytotoxic T cells will bind with the MHC class 1 protein, and the CD4 co-receptors on helper T cells will bind MHC class 2. So 8 to 1, 4 to 2. On the right-hand side, you can see a visual showing you the MHC complex. We have the alpha 1, 2, and 3 subunits. We have the beta, we have the antigen fragments, and over here on the T cell membrane, you can see there's an interacting portion of alpha and beta chains that will lock onto the receptors. So CD8 co-receptors go to MHC class 1, CD4 co-receptors go to MHC class 2s. Over here is an example of a cartoon image showing you the docking. So the CD4 interacts with the MHC class 2, CD8 interacts with MHC class 1. That is very important to remember. 8 to 1, 4 to 2.